KURINJI Senior Secondary School, CBSE, Namakkal.
Successful result producing school in state every year.
Administered by the Board of Directors with over 35 years of experience in the teaching field.
Special attention on art, craft, music, dance, silambam, yoga, skating and karate.
Focus on Hindi Sabha and Spell Bee Olympiad exams.
Smart class for effective and innovative learning and well-furnished library.
Solitary importance on sports and games in state and national level.
WhatsApp facilities to know activities of students and school inside the campus.
NEET, JEE, IIT coaching experts from Rajasthan and Andhra Pradesh along with our effective teachers.
Admission registration for classes LKG to +2.
KURINJI Educational Institution, Kavetipatti, Namakkal. Admission in progress. CBSE, KG to 12, contact 9025895176. Metric 6 to 12, contact 9344567484. For online admission, log on to www.kurinjischoolnkl.in.
In this video, we are discussing continuation information of what is the chapter? No, cell the unit of life. So here also maximum information comes under introduction. So why? Because in previous video along with secondary growth I have started to give information or introduction of cell the unit of life chapter. No, so in this particular video also information but introduction and primary information. So but important though. So definitely suppose important points not there means I will not give I will not make video, no, so thereby so these points also important, listen carefully. So now let us move forward to learn so one by one. Now cell theory. So in previous video I have given a small introduction regarding cell theory. No, cell theory, Sladen and Swan, they have been proposed. So likewise continuation information. Ipo Sladen botanist, German botanist, and Swan British scientist, zoology scientist. Likewise in previous video few points we learned. No, likewise here continuation ma. Okay, Swan also concluded. Yare Swan. Adhe ma rendu scientists is there, no, Sladen one and Swan another one, no, it is the Swan. Okay, Swan also concluded. Ipo also concluded means munnadi edo discovered paniyachi. So what is that? So he was experimented or he was observed different types of animals, and what he was given information, so that is so what he was given information, so animals definitely consist of delimiting structure with cell membrane, he was given information. Next one, now Swan also concluded based on his studies on plant tissues. Amma, here did you observe one point? So actually Swan belongs to zoology. I'm a zoologist, but here he also concluded what? Based on his studies, means he was studied or experimented on even on plant tissues also and present, and when he was observed on the plant tissues, no, so he is giving one conclusion is that so the presence of cell wall is a unique character of plant cells. Very, very important point. Actually, even though he is a zoology scientist, so a geologist, so what is the information he was given to us? So definitely plant cells are having a unique character, that is possessing of cell wall. So presence of cell wall is a unique character of plant cells. Next one, on the basis of this one, okay, on the basis of this, Swan proposed the hypothesis is that the bodies of animals and the plants are composed of cells and products of cells. This is important. So maximum points are important only. Now here on the basis of this one, so here what is the information Swan was given? So he was experimented on the animals, and he has given information delimiting structure of animals is cell membrane. He said like that. And one more important point, and he was experimented on the plant tissues and concluding that outermost delimiting structure, delimiting structure velida, what is a layer which is situated outside, you know. So that one, our subject point of view, we will be calling by one more name, delimiting. Sarima, so means in plant cells, no, outermost protecting layer is a cell wall. In animals, outermost protecting layer is a cell membrane. cell membrane or plasma membrane. So try to remember these 2 points. Next one, finally, what is information is giving on this basis. So one proposing one kind of a hypothesis. So there in this particular hypothesis he is giving information is that, so what is that? The body of animals and the body of plants is composed of cells, composed of cells and the products of cells. Okay, so thereby here, so what are the living organisms are there on the globe? No, all living organisms means either the plants or the animals definitely composed of cells and the products of cells. கொஞ்சம் பத்திரமா நல்லா பார்த்துக்கோங்க இது. So thereby you will save these points in your mind. Next one, Sladen and Swan together formulated the cell theory. இப்போ ரெண்டுமே together, okay, not separately. So Sladen as well as Swan together formulated cell theory. So this theory, however, did not explain as how new cells were formed. So means here just they have given information. So one scientist, botany scientist and another one zoologist, they have experimented on the plants as well as animals, so they have proposed cell theory. So they have proposed the cell theory, but what is the point? How these new cells of formation taking place, they didn't explain. But here what they have observed, no, new cells of formation taking place they have observed, but how these new cells of formation taking place they didn't observe or they didn't explain at the time. Rudolf Virchow, this scientist name, in 1855. Rudolf Virchow in 1855 first explained that the cells divided and new cells are formed from the pre-existing cells. This is important. So thereby, actually two scientists together formulated cell theory. So two scientists names are Sladen and Swan, but here they didn't give information or the explanation regarding how new cells of formation taking place. I hope you understood. Next one, so now Rudolf Virchow for the first time explained. So what he was explained for the first time, he explained that cells are divided and new cells are formulated or formed from the pre-existing cells. இது important point. One or two times கொஞ்சம் நல்லா revise பண்ணிக்கோங்க. Okay, so one more time, the cells are divided and new cells are formed from the pre-existing cells. Pre-existing cells na already அங்கே cells இருக்குன்னா அது pre-existing cells. So from those existed cells, suppose if a new cells of formation taking place, so that kind of information was given by Rudolf Virchow. Pre-existing cells means already irukku cells. So what are the cells or present already? Our subject point of view, or simply we will be taking as a pre-existing. Pre means munnadi only, no? So pre-existing cells. So these pre-existing cells involved in the development or the formation of new cells. So by the process of cell division. I hope you people understood this. Next one, so when the new cells are formed from the pre-existing cells, so that particular thing in I think so in another language point of view, so we will be calling as omnis cellula e cellula. So that is omnis cellula e cellula. Okay, means here new cells of formation taking place from the pre-existing cells. So instead of calling off new cells of formed from the pre-existing cells, instead of writing this entire information, suppose if you write omnis cellula e cellula, that is brings the information about the statement. I hope you understood. Means now omnis cellula e cellula means what? New cells are formed from the pre-existing cells. Omnis cellula e cellula means new cells are formed from pre-existing cells. Next one, he modified the hypothesis. He means here Rudolf Virchow. He modified the hypothesis of Sladen and Swan to give the cell theory a final shape. Actually cell theory proposed by Sladen and Swan, they have together formulated cell theory. Rudolf Virchow, he modified the cell theory. He modified the cell theory and he would like to give final shape to the cell theory. That's why he modified the hypothesis of Sladen and Swan. Modified the hypothesis of Sladen and Swan to give the which one to give the cell theory a final shape. So thereby final shape of cell theory was explained by or given by Rudolf Virchow. Next one, cell theory as understood today. So is that, so means today the cell theory, so we can understand like this. How? So two statements are mainly or important in cell theory. So what are those? No, all living organisms are composed of cells and products of cells. Amma, one more time. All living organisms are composed of cells and products of cells. Emma, so means this particular point here we have learned now once you see. So cells or the animals or the plants are the composed of cells and the products of cells. So here that information we are learning under hypothesis or under the cell theory, modified cell theory. Okay, now here their first statement is all living organisms are composed of cells and the products of cells. Next one, all cells arise. All cells are simply are formed also we can use. All cells are formed from pre-existing cells or all cells arise from pre-existing cells. So this particular point also try to remember. Next one. So an overview. Let us have a look on the overview of a cell. So suppose if you take one particular cell, so there what are the main important points to be covered or what are the components are present within the cell, everything now we can focus. That's why side heading is an overview. So complete information, not konja information. Complete information. That's why overview of a cell. Now a onion cell. Onion cell we know very well. No, mummy will cut onions in our kitchen. So onion cell if you take there, no, which is a typical plant cell. So it is best example of a particular plant cell. So and has a distinct cell wall. Why? Because a plant cell, no, now only we see no. So plant cell means definitely one unique character is there, that is the presence of cell wall. Now only we see it. So thereby in onion cell, no, suppose if you take that particular onion cell as a typical or the example of a plant cell, so distinct cell wall is present. That particular distinct cell wall as its outer boundary. Outer boundary. So that particular outermost delimiting structure or outer layer or outer boundary. Likewise, many terms we can use. Now here, so that particular thing just below it, once you see, and just within it, it is known as cell membrane. Means here just below the cell wall one more layer is present, that is cell membrane. Means here if you take in comparison of plants and animals, what is the main important point we have to discuss? No, cell wall is present as outermost delimiting structure in case of plants. And in plants point of view, below the cell wall we will be finding the presence of cell membrane. My dear students, important. So below the cell wall in case of plants, no, below the cell wall what we can find on? Cell membrane. But in case of animals, suppose in case of animals, no, outermost protecting layer only is cell membrane. There is no chance to the presence of cell wall in case of animals. So nalla purinjuko. So next one, so here next information, next point, the cells of the human cheek. Human cheek means definitely we are all comes under animals only, no. So human cheek have an outer membrane as the delimiting structure of the cell. So already we have discussed so many times. So thereby in animals point of view or in human cheek point of view, if you take there, no, their outermost delimiting structure is outer membrane or cell membrane. Next one, inside each cell a dense membrane. One more time, inside of each cell a dense membrane bound structure is present. That particular dense membrane structure or dense membrane bound structure, our subject point of view we are calling as nucleus. Once you see inside the cell a dense membrane bound structure, a dense membrane bound structure is known as nucleus. Okay, this nucleus contains chromosomes, that's we know very well. So why? Because within the nucleus only we will observe the presence of genetic material, and that particular genetic material is present in the chromosomes. So that information in our childhood studies only we have learned. What is the point now I am going to give you is, first of all, delimiting structure in case of plant cells is a cell wall, and within the plant cell wall so second layer is a cell membrane. Next one more layer also is there. One more structure also is there with membrane. Membrane bound structure means for nucleus also membrane is there. Membrane bound structure is present. Next one, within the nucleus chromosomes are there, and which in turn contain genetic material. Which in turn contain genetic material. So thereby, my dear students, what is the point? Within the nucleus we are observing the structures of chromosomes, and on the chromosomes or within the chromosomes we are observing the presence of genetic material. So in this particular side, so what is the information we have learned? So Swan, Sillidan and Swan together proposed cell theory, but they didn't give explanation how new cells of formation are taking place. But Rudolf Virchow for the first time explained. So what is the information he was explained? So Rudolf Virchow explained that new cells are formed from the pre-existing cells. So that is only simply we will be calling as omnis cellula ucellula. Next one, he modified, means Rudolf Virchow modified the cell theory and given a final shape. So according to that final shape of the cell theory point of view, all living organisms are composed of the products of cells, and definitely all cells are formed from the pre-existing cells. So that is information. Overview point of view. So what are the points? So if you take a plant cell, outermost protecting layer cell wall is there, and if you take animal cell, outermost protecting layer cell membrane is there. But in plant cells point of view, outermost protecting layer cell wall below one more layer we are finding, that layer is cell membrane. So that is information. This side will know. That side so what is information? So within the plant cells or within the cells, any particular cell, that is maybe either a plant cell or animal cell. So within the cell, definitely we will be finding of dense membrane bound structure. That particular structure only our subject point of view calling as nucleus, and within the nucleus we are finding the presence of chromosomes. So on the chromosomes or within the chromosomes, or these chromosomes contains what is the information? Genetic material known as DNA. So why? Because we will have a genetic material DNA only, Adida. Next one, cells contain, or cells that have a membrane bound nuclei are known as eukaryote or eukaryotic. Means so suppose if the cells, or what are the cells that have a membrane bound nucleus? One more time you see first line. Cells that have membrane bound nucleus. So suppose if the cells consist of membrane bound nucleus, cells with nucleus, that nucleus also with boundary. That nucleus also with membrane. Membrane bond is there, means that particular nucleus or that particular cell we will be taking as eukaryotic cell. Point you understood? A simple thing. Cell with cell ulle nucleus surrounded by the membrane. Nucleus surrounded by the membrane means that particular cell we will be calling as eukaryotic. Actually eukaryotic meaning EU here, EU is there, meaning true, and karyo means nucleus. Okay, so means true nucleus is present. True nucleus is present means when we will be calling as true nucleus cells means, suppose if the nucleus is bounded with membrane. So suppose if the nucleus superly bounded with membrane, that particular cells we will be considering as eukaryotic cells. And whereas cells that lack of or lack a membrane bound nucleus, here within the cells nucleus will be there, but that particular nucleus is not surrounded by membrane. Okay, means here what is the information? So here prokaryotic, that particular cell we will be taking as a prokaryo. Pro means before, or our subject point of view we will be taking as meaning as a premature or unmatured, easy word. Okay, unmatured means Karyo means nucleus and pro means premature or undeveloped one. So means when we will be calling that particular words, suppose if the membrane bound is there around the nucleus, means we are calling as eukaryotic, well developed one. And around the nucleus, so suppose if the membrane bound is absent, means that particular cells we will be taking as prokaryotic cells. I hope you understood this one. Next one, in both prokaryotic organisms as well as eukaryotic organisms, or prokaryotic cells and eukaryotic cells, a semi-fluid matrix. One more time, in both prokaryotes and eukaryotic cells, no, we will be finding. So what we will be finding? No, a semi-fluid matrix. Semi-fluid matrix is present. So that particular semi-fluid matrix we will be taking as cytoplasm. So means within the cell, what is the liquid we will be finding of? So the liquid which is present within the cell, no, we will be considering as cytoplasm. So cytoplasm in information point of view, so semi-fluid matrix which is situated within the cell, we will be considering as cytoplasm. So that particular cytoplasm occupies the volume of the cell. So occupies the volume of the cell. So occupies the volume of the cell. Next one, the cytoplasm is the main arena of cellular activities. Semi-fluid matrix is there, no? This particular semi-fluid matrix, cytoplasm, occupies the volume of the cell. Occupies the volume of the cell. Occupies the volume of the cell. What is the main important use of this one? So this particular cytoplasm is main arena of cellular activities. This is the important point. In so many examinations they have given. Means, so what is the main arena? Means what arena means place. What is the main place for cellular activities in the cell means? So we must remember the cytoplasm. So cytoplasm is a main place. This arena no main place. So site, main place means likewise we will calling no. So likewise, cytoplasm is a main arena of cellular activities in both the plants and animal cells. Why? Because already we have seen the presence of cytoplasm in both prokaryotes and eukaryotes, isn't it? So thereby here what is the point? In both prokaryotes and eukaryotes, cytoplasm is present. So this particular cytoplasm occupies the volume of the cell. Later this particular cytoplasm is mainly involves in the all cellular activities in both both plant cells as well as in animal cells. Next one, various chemical reactions. Various types of chemical reactions occur in it. So there are different types of chemical reactions. So there are so many number of chemical reactions occurs within it or in it. So in it means, so definitely there are different types of chemical reactions takes place in the cytoplasm. Sir, why sir here so many kind of chemical reactions are taking place? Means to keep the cell in the living state. Okay, means when the chemical reactions are taking place within the cytoplasm, no, so then only because of the chemical reactions only that particular cell will be in living state. My dear students, so try to understand. So because of the presence of or because of the activity of chemical reactions, so that particular cell will be in living state. So means all chemical reactions or all metabolic activities, definitely at the site of all metabolic activities or the chemical reactions is cytoplasm. It is the main site. So try to understand. Later, what are the main important points? No, beside to the nucleus. Beside of or beside the nucleus, the eukaryotic cells have other membrane bound distinct structures. Here one important point. So beside the nucleus means where only in case of eukaryotic cells. In eukaryotic cell, along with or beside to the nucleus, there are different membrane bound distinct structures also are there, or the special structures also are present. So these particular structures we will be calling as organelles or cell organelles. So cell organelles are present. So these organelles names are ER, endoplasmic reticulum, GC, Golgi complex, lysosomes, mitochondria, microbodies and vacuoles. Still so many are there. So while taking a class, definitely I will give you one by one. So endoplasmic reticulum, Golgi complex, lysosomes, mitochondria, microbodies, vacuoles. Okay, next one, the prokaryotic cell lacks such membrane bound organelles. My dear students, very important point. What is that? Along with the nucleus in eukaryotic cell, no, all these cell organelles are present. But in case of prokaryotic cell, if you take or if you observe the prokaryotic cell, there lack of such membrane bound cell organelles means all these structures are absent. Where? In prokaryotic cell. So in a prokaryotic cell, so what are the cell organelles? Male we have seen, no, LMA absent in case of prokaryotic. Lack means absent only, no. Okay, so that is the point. Now ribosomes are so here few exceptional points are there. Listen carefully, these points only are backbone. Now here you see, so ribosomes are non-membrane bound cell organelles found in all cells of both prokaryotic as well as eukaryotic organisms. So my dear students, what is the point? No, so male we seen in eukaryotic cell, no, there are different types of cell organelles are present, but in a prokaryotic cell, no, LMA absent. Now only we seen, no. Now here one important point, that is ribosomes. Ribosomes, once you see, these are all ribosomes are non-membrane bound cell organelle. Non-membrane bound cell organelles means membrane means ribosomes are surrounded by, so we could not see the presence of any membrane. So that's why non-membrane bound cell organelle. So means here this particular cell or these particular cell organelles are present in all kind of cells. All cells means both prokaryotic cells as well as eukaryotic cell. Means here, my dear students, so one point try to understand panikonga. What is that? In prokaryotes, all type of cell organelles are absent except ribosomes. Why? Because now only we are observing, no, male, what is the point? We seen all these kind of membrane bound cell organelles are absent in a prokaryote we seen. What is the point? Ribosomes also non-membranous structures only, no. These kind of non-membranous structures are present in both kind of cells, eukaryotic or eukaryotic cells as well as prokaryotic cells. So in prokaryotes, no, definitely we will find out the presence of ribosomes. Here one more point in the next classes I will give you. In ribosomes, in prokaryotes, no, ribosomes type 70S type. Just voice mattum try to remember panikonga. And in eukaryotes, no, type of ribosomes 80S type. So next one, what is information or what are the subunits or what is the function of ribosomes, everything. So in future classes you will learn. Now within the cells or within the cell, ribosomes are found not only in the cytoplasm. So thereby here we are understanding the location of these particular ribosomes. If once you see, so within the cells ribosomes are found not only in the cytoplasm, means cytoplasm nalli kandipa irukku, and not only in the cytoplasm but also present in the two cell organelles known as chloroplast and mitochondria. And one more is on rough endoplasmic reticulum. So means here definitely ribosomes are present in the cytoplasm. Along with the cytoplasm, we will observe the location or we will observe the presence of ribosomes in two cell organelles. One is chloroplast in case of plants and in mitochondria in both plants and animals. And not only these two cell organelles, we will be finding on rough endoplasmic reticulum also. We see endoplasmic reticulum, no, rough endoplasmic reticulum, because of the presence of ribosomes on this endoplasmic reticulum, rough endoplasmic reticulum becomes rough. One more time, rough endoplasmic reticulum is there, no. This endoplasmic reticulum, because of the ribosomes, converted into rough endoplasmic reticulum. Suppose when ribosomes are present on the endoplasmic reticulum, no, now we will be calling that particular endoplasmic reticulum as rough endoplasmic reticulum. Suppose if ribosomes are absent on the endoplasmic reticulum, means what we can call? Can you guess it? Smooth endoplasmic reticulum. Means thereby what is the difference we can find out? Rough endoplasmic reticulum and endoplasmic reticulum consists of ribosomes. Smooth endoplasmic reticulum means endoplasmic reticulum not consist of ribosomes. That is only no meaning. So means now here one point let me highlight, that is Ribosomes are present in both prokaryotes and eukaryotes, but difference irikama. Next one, these ribosomes not only present in the cytoplasm but also present in the chloroplast as well as mitochondria. Chloroplast is present in the plants. Why? Because chloroplast only absorbs the sunlight and helps in the process of photosynthesis. In a photosynthesis chapter, I'll explain the entire information regarding the chloroplast. Next information point of view, animal cells. Animal cells contain another non-membrane bound cell organelle called centrosome. Ipo animal cells only in animal cells one more non-membrane bond, means here in ribosomes are present in both in all kind of cells, prokaryotes, eukaryotes, in eukaryotes plants and animals, in all cells we will be finding the presence of ribosomes. Ipo in animal cells point of view, in animal cells one more another membrane bound cell organelle is there. Another membrane bound cell organelle is there. That particular another membrane bound cell organelle is centrosome. So thereby, my dear students, so suppose if I ask a question like this, in animals total number of non-membrane bound cell organelles, so can you guess how many? Rendu. Amma, very good. So one is ribosome and one more is centrosome. So try to understand. So then sir, in animal cells non-membrane bound cell organelle centrosome is there. What is the use of this centrosome, sir? Means once you see which helps in cell cycle, which helps in cell division. One cell, rendu cell, 4 cell. So at the time centrosome definitely helps animal cells to divide. So that's why definitely, so centrosome helps in cell division. Next one, cells differ greatly in size, shape and activities. Cells in anatomy we have learned there are different types of cells are there: parenchymatous cells, cholenchymatous cells and sclerenchymatous cells. Function, activity, shape, everything is different. Even activity, everything is different. Size point of view, shape point of view, activities point of view, difference we found it or not? We found, so for example mycoplasma. Mycoplasma is smallest living cell. Listen carefully. So this particular mycoplasma cell we will be considering as smallest cell. We will be taking as a smallest cell are only 0.3 micrometer in length. So here the mycoplasma size is, so just our length is 0.3 micrometer in length. So that's why this particular mycoplasma we will be considering as smallest cell. And whereas in case of a bacteria point of view, bacteria that could be 3 to 5 micrometer in diameter. So length of the bacteria or the size of the bacteria is 3 to 5 micrometer, whereas mycoplasma point of view just 0.3 0 point I think it is not appearing clearly, but 0.3 micrometer in length. So that's why here this particular mycoplasma we will be considering as the smallest, actually here in a textbook living but smallest living cell we can call. Okay, smallest cell or the smallest living cell. Okay, so still continuation information is there in the next video, let us see. But in this video, what is the information we have seen now? Entire information is introduction as well as to understand the remaining concepts. So this entire information is backbone. So if you could not understand by first time, so one more time try to watch. So, okay, so learn the information, so be in a good position in our subject. Take care and be safe at home.